Morning students, we are in the lesson plant tissue culture. We have seen about the techniques involved in the plant tissue culture in the previous classes, how to maintain the aseptic environment, how to sterilize the room and how to sterilize the media and the media preparation, MS media, the mursage and murasage and skoog medium, all these things, and what are the pH level, temperature, how to aerate the room, then inoculation of the culture. These things we have seen in the previous classes, and also we have talked about the types of the plant tissues cultures. We have seen talked about the organ, meristem, protoplast, cell culture. Till this we have completed in the previous classes. Now we'll move to the one cultures. We'll start with the organ culture. We'll start with the class of the organ culture. What is organ culture? The culture of the embryos, anthers, ovaries, roots, shoots or other organs of the plants on the culture media. Here we are going to take the embryos, the young one, and then the anthers, ovaries. All these things are isolated and they are grown in the culture media. They will give rise to the plantlets, small plants, and from that we are going to transfer that to the normal environment. Okay, that is, we are taking the embryos, that is called as organ. Organ means what? Parts of the plants. We are taking the parts of the plant and they are grown in the culture media. The parts of the plant, they are isolated. The cells from the parts of the plant, they are isolated and they are grown in the culture medium, and that culture medium will form the plantlet, new plants, and from that they are transferred to the natural medium. That is called as organ culture. The next thing is the meristem culture. Meristem means the culture of any plant meristematic tissues on the culture media. Any plant meristematic tissue, it might be any part of the plant where the meristematic tissues are present, that is taken out and that is grown in the culture media. The picture is also shown here. See, they are taking the shoot system, the leaves, older leaves, that is removed, and then also the shoot tip. See the larger leaves and the shoot tip with the growing tip after removal of the enclosing leaves. The plantlets are formed. Next is the protoplast culture. What do you mean by protoplast? Protoplast are the cells that have cell membrane but not the cell wall. They will not have cell wall, but they are bounded by the cell membrane. Using the protoplast culture, it is possible to regenerate a whole plant from a single cell. Okay, from the protoplast, a whole new plant can be formed, and it will form the somatic hybrid. Somatic hybrid, and this will involve some. This have involved some certain steps. First, what is protoplast? Protoplast are the cells without the cell wall, and it has got the cell membrane or the plasma membrane. Using the protoplast, it is possible to generate a new whole plant from the single cell. Using the protoplast, you can grow a single new plant. That involves some techniques. First one is the isolation. Isolation means removal. We are taking out the protoplast from the plant. How it is done? We'll go by the rules. Some small bits of the plant tissue like leaf tissue are used for the isolation. What they are taking is bits of the plant tissue, like they are taking the leaf tissue. For example, they are taking the leaf tissue. These are bits of the plant. They are isolated, they remove, and that tissue, what they are doing, they are cleaning, then they are immersing in some solutions. That solution name is the tissue is immerse. Immerse means that is dipped. That is dipped in 0.5 microzyme and 2% Onazuka cellulase enzyme. That is dissolved in 30% sorbitol or mannitol at the pH of 5.4. We have told we are maintaining some temperature, pH, all these things are made like that. The pH for the protoplast isolation is 5.4, and then it is then incubated overnight. It is incubated in the incubator at overnight for 25 degree. After the teasing of the cells, after the removal of the cells, the protoplast are obtained. After cleaning, everything is done, the protoplast is obtained, and then it is transferred to 20% of sucrose solution to retain the viability. After it is dipped in the immersed in the solutions of sorbitol and onazuka and incubated, it is again transferred to 20% of sucrose to test the to get the viability. For viability was live cells. Then they are centrifuged. Centrifuge is nothing but it is an instrument in which the cells, they are made to circulate in a direction centrifugal force applied, that is applied, and it is made to move to get pure protoplast as different from debris of the cell wall. The dead cells are removed and the pure protoplast is got. It is done by the centrifuge. Centrifuge is a machine in which centrifugal force is applied. From that, when the force and the solution and the test tube is made to circulate in a centrifugal force, and then we will get the pure protoplast without any dead cells. Next step is the fusion. Fusion, it is done through the use of suitable fusogen. The fusogen here used is PEG. PEG is nothing but polyethylene glycol. So fusion of protoplast is done with the help of the fusogen, that is called as polyethylene glycol, and that is incubated at 25 to 30 degree. The concentration of pegged with calcium 2+ ions, and then this protoplast, when that is applied with the polyethylene glycol and it is incubated along with the calcium 2+ ions, it forms agglutinations. Agglutination is nothing but formation of groups or the clumps. When it is applied, they will form group like this. They will form group that is called as clumps. They are called as clumps of cells and they fuse. And the next step is the culture of protoplast. First thing we are isolating, second thing they are fusion of protoplast is done, and next is the culture of the protoplast. The MS liquid medium, MS is Murasage and Skoog medium. In that medium is used with the some modification in the droplets and plating or micro dripping. It might be done like plating means streaking out, the circular plate is taken out. In that it is streaking is like this, or the plating, or a drop of the micro drop array technique is used, or droplet of the cell is protoplast is dipped or dropped into that plate. The protoplast viability is tested with fluorescent diacetate before the culture. Before the culture it is tested. It is tested with the this type of chemical fluorescent diacetate. The culture is incubated. It is incubated in the continuous light 1000 to 2000 LUX at 25 degree. Then the cells with the cell wall formation. First, the protoplast will form the cell wall within 24 to 48 hours. After it is formed the fusion, then it is taken in the MS liquid medium. Either the droplets or micro drops are used, techniques are used. In that it is tested with the fluorescent diacetate, then it is incubated. After it is incubated, the cell wall formation occurs with the protoplast at within 24 to 48 hours. And the first division the first cell division new cells occurs between 2 to 7 days. In 24 to 48 hours, in 2 days cell wall formation occurs. After that the new cells will occur in the 1 week, 2 to 7 days of culture. So I repeat, first one is the isolation of the protoplast, and then in that isolation we are dipping in some solutions, the different solutions, macrozone, onazuka, mannitol, and incubated, and then it is viability is tested. Test the viability, it is dipped in 20% of sucrose, and then it is centrifuged to get rid of the dead cells. And then the fusion is done with the fusogen. Fusogen name is polyethylene glycol, again incubated, and calcium with the presence of calcium 2+ ions, they form agglutination. Agglutination is the formation of clumps or groups. Then they form the fusion. Then it is taken in the culture medium and it is tested with the fluorescent diacetate and incubated 1000 to 2000 degree. Then After incubation in 24 to 48 hours, the cell walls are formed, and then in 2 to 7 days the new cells arises. After that, selection of the somatic hybrid cells are done. The fusion products of the protoplasts within the nucleus, without nucleus. First they are formed, they are without the nucleus, no nucleus is formed. First no nucleus is formed within the protoplast, that is called as hybrid. The cells without the nucleus, they are called as hybrid. Following this, after that hybrid is formed, nuclear fusion happens, and that is called as somatic hybridization. That is called as somatic. What is somatic hybridization? The protoplast, the fusion of the protoplast without the nucleus, they form this hybrid. After hybrid formation, they form the nuclear fusion, and then that is called as somatic hybridization. The next topic is starting with the cell suspension culture. The growing of the cells, including the culture of the single cell or small aggregation of the cells in vitro. It is grown inside the test tube in the liquid medium, that is known as cell suspension. Cell suspension means what? The cultures, the cells, the culture of the single cells or the aggregate of the cells, or aggregate means what? A group of the cells. These cells, they are grown inside the test tube in the liquid medium, that is known as suspension. Suspension means what? It is in the liquid medium. The suspension, it is prepared by the portion of the callus, a part of the plant that has to be transferred, that is known as callus, to the liquid medium, and it is agitated. Agitated means what? It is shaked. It is shaked with a rotatory shaker instrument. The cells are separated from the callus tissue and used for the cell suspension. From the tissue, for example, a part of the leaf is taken. From that, the callus tissue is separated, and from the tissue the new cell suspension is formed, that is in the liquid medium. Now, production of secondary metabolites. This paragraph it is telling about the plants. Certain plants, they will produce some metabolites that are not useful for the plants, but they are useful for the humans or any other thing. That is called as the secondary metabolites. The metabolites that are produced by the plants that are not useful for the plants, but they are any other way useful to the human beings. For example, the cell suspension culture useful for the production of secondary metabolites like alkaloids, flavonoids, terpenoids, phenolic compounds, and recombinant proteins. They are the chemical compounds that are not required by the plant for normal growth of the plant and development, but they are produced as a byproduct. For example, see isolation of indole alkaloid from Catharanthus roseus plant cell culture, and the process of the production of secondary, they can be scaled up, automated using bioreactors, and by many strategies like biotransformation, elicitation, and immobilization have been done used to make the cell suspension and more efficient in the production of secondary metabolites. To produce the secondary metabolites, we are using biotransformation, elicitation, and immobilization. We are not going to move the thing. And then see, for example, it is given in the tabular column, digoxin. See, if you take digoxin, it is in a medicine that is used to cure the cardiac. It is used as a cardiac tonic for and it is taken from the digitalis purpura. This is from the digitalis purpura, digoxin. And codeine is nothing but it is given in the allergies, and then for cough syrup it is taken from Papaver somniferum. Analgesic, and capsaicin, that is capsicum, the green chili or the red chili, it is taken from that plant. It is used for the rheumatic pain treatment. It's given as a ointment. And vincristine, Catharanthus roseus, anti-carcinogenic. And then quinine for malarial treatment, Cinchona officinalis, that is used for the malaria treatment. So these are secondary metabolites. They are produced by the plants, but they are not useful for the plants, but they are useful for the human beings. So to produce the secondary metabolites, the strategies used are biotransformation, elicitation, and immobilization have been used to make the cell suspension, and that is used in the production of secondary metabolites. They are automated using bioreactors. Okay, I think you should know. And next is the plant regeneration pathway. from the explants, that is removed tissues, small tissues, the plants can be regenerated by somatic, embryogenesis or organogenesis. This is shown as a flow chart. See, from the explant it can be direct embryogenesis or indirect embryogenesis. This is the pathway for the regeneration, plants regeneration. That is also given as with the help of the plant structure. It is removed and then the somatic embryo is formed. How the shoot root organogenesis, that is known as indirect, and from that rooting the new plants are formed. From direct organogenesis also new plant is formed. It might be either due to indirect or direct. Next is somatic embryogenesis. Somatic means the cells apart from the gametes. Somatic embryogenesis is the formation of the embryos from the calyx tissue directly through embryos are called embryoids. When the somatic embryogenesis are formed through the embryos, they are known as embryoids, or in vitro cells directly from the pre-embryonic cells. They are differentiated. They differentiate into embryoids. See, somatic embryogenesis means what? The embryo formation from the tissue directly from the embryos, that is known as embryoids, or in vitro inside the test tubes from the pre-embryonic cells that differentiate into embryoids. Applications, what are the uses? Somatic embryogenesis provides potential plantlets. The plantlets that are produced they are very potential. Their hardened period can be established into the plants. After that, they will form the new plants very easily. Somatic embryos can be used for the production of synthetic seeds, artificial seeds, and then somatic is also now reported in many Allium sativum, all these plants, Oryza sativa, Horridum vulgare, Z maize, and this is possible in any of the plant. What is synthetic seeds? These seeds are produced by the encapsulation of the embryoids in agarose gel for calcium algae or calcium alginate. That is, the seeds produced they are encapsulated of the embryos in agarose gel. We are keeping them in the agarose gel, agar gel. Okay, that is known as the somatic embryogenesis. Next is organogenesis. Organogenesis means the morphological changes occurs in the calyx that leads to the shoot and the root. See, that occurs in the shoot and the root. These organ formation inside the shoot and the root, that is known as organogenesis. Okay, the calyx that is differentiated into shoots and roots. When it is differentiated into shoot, that is known as calogenesis. That is differentiated into root, that is known as rhizogenesis, and they form the plantlets. Organogenesis they may be induced in vitro also by producing the plant in regulators in MS medium. MS medium is, I am repeating all the time, that is the Murasage and Skoog medium. Then auxins and cytokinesis, that are growth hormones. You might have learned in your previous classes, auxin and cytokine, they are the growth hormones of the plants that induce the shoot and the root formation. Auxin and cytokinase. And all these things are done in the, we are talking about the organogenesis, and also we have seen about the somatic embryogenesis. Next class we will see about the applications of the plant tissue culture.